For infectious diseases, it's very important that we have a very narrow understanding of the infection process and the pathogen that causes this infection as early as possible so we can intervene with optimal treatment. My name is Eleftherios Mylonakis. I'm the chair of medicine at Houston Methodist, and I also have a research lab and a group that studies antimicrobial drug discovery and new diagnostics. What was fascinating to me is why those pathogens that are surviving in nature, why they developed their virulence mechanisms against humans. Those pathogens, those fungi and bacteria, they developed those traits of virulence when the humans and mammals in general were a very small part of the biomass. It makes sense that they developed those virulence traits in order to survive in the environment, to survive when they interact with amoebae, when they interact with insects or microscopic worms. And then it so happened that those same virulence factors can have an effect during the infection process of mammals and humans. My thinking was, if I go back and we try to study those environmental microorganisms, then we can go to the core of their virulence and find a simple model host where we can study them, and also study how insects and how worms and how amoebae are able to interact and control those pathogens. And maybe that can give us some tools in order to treat our patients. There is a lot of complexity in understanding the nature of antimicrobial resistance. It has to do with overprescribing antimicrobials, overusing antimicrobial agents, also with the human population where we have a higher number of immunosuppressed individuals or individuals with immune system that is manipulated medically, such as transplant recipients, and climate change that also could be contributing as a factor. Then we have other developments such as the movement of population. We saw that happening very quickly with the COVID-19 pandemic, and we are probably witnessing it in a more slow but equally concerning manner to play out with the antimicrobial resistance. We want this new area of discovery to bring together the medicine and engineering in order to develop new diagnostic technologies, to develop new biomaterials that are more resistant to infection. We want this treatment to be very narrow so it doesn't affect the other microbes in the microbiome of our patients and it doesn't promote resistance. Bioengineering will help answer a lot of those questions, help answer those challenges. At Houston Methodist, we have the first class of bioengineers, medical students who graduate with an MD and also a master's in engineering. The opportunity that we have is that engineering with AI, with in silico research, with new biomaterials is ready to flourish, is ready to grow. The challenge that we have is putting together engineers with biologists and clinicians. And hopefully Houston Methodist will be an umbrella where those two disciplines can come together and develop the discoveries for tomorrow. Texas Medical Center provides a unique opportunity to collaborate with different disciplines. And especially Houston Methodist allows for the Research Institute and the hospital to be under one entity. If we take advantage of this efficiency, we can take discoveries from the bench to the bedside as soon as possible. That will have a meaningful effect to our patients because we can provide our patients with choices years before they go through all clinical trials approvals. It takes about 10 or more years for a lead compound to make it to clinical practice. We want to make Houston Methodist the place for outside discoveries to be evaluated. We want to provide a seamless transition between discovery to a clinical practice and provide those options to our patients. I think that translational research environment is quite unique here, and it's a unique place to do research and to try to navigate discovery from a hypothesis all the way down to clinical trials in the same area. What happens a few times in a scientist's career is that you have a complete understanding of the phenomenon. You feel it in your heart. You feel the experiment, you feel the process. You have a feeling and an understanding of the next step, and you can see it through.